Yes, my name is Brad Larkin, and I was going to talk to you today about intermediate and advanced Y-DNA topics, things that are not beginner stuff. And I think most of the group on here is pretty experienced. So tools and techniques, some of the tools I've built, some tools I use, other people's, but to try and go beyond the basic set of tools. So we're going to kind of do a deep dive into Y-DNA Next Gen testing for tests like Big Y-700 and YSeq WGS product. We're going to go from biology to data to the phylogenetic tree to geographic interpretation. So the topics are going to include raw read alignments to the genome, which it produces your SNP results, understanding your terminal SNP and why it changes, how to use novel variants, unnamed variants to extend the phylogenetic tree of your family, and tips on getting the most info and avoiding bugs in current tools using real-world examples. Part one is SNP detection. Part two is going to be trees and interpreting SNPs, collections of SNPs. And part three is some tips and things. So our goal here is for you to have a deeper understanding, learn about the existence of visual tools for reviewing Y-DNA SNP results, and understand the concepts of terminal SNP and phylogenetic tree. The intended audience of this is usually Y-DNA project administrators, aficionados, and independent researchers, those trying to find new branches, new mutations, and also genetic genealogists and test takers who want to get a deeper understanding, and people who are building DNA software and tool builders. The lab conducts these kinds of tests that we're looking for here include family tree DNA, YSeq, Full Genomes Corp, 23MoFang, and Nebula Genomics. So this presentation is not intended for adoption solving, criminal or missing persons investigations, or DNA tests involving less than 2 million base pairs. So that's Ancestry DNA, 23andMe, Living DNA, MyHeritage. If you've only got one of those tests, you want to apply the things here, you would need to upgrade or order one of those tests on the previous slide. This little illustration here in the bottom left, I got it from Nebula Genomics, but it shows how much more DNA is being sequenced compared to the microarray test by 23andMe and stuff. They're on the top. The bottom is what whole genome sequencing test goes. So you can see it's much, much more information. So some background on myself. I'm a U.S. Marine Corps veteran. I tend to pronounce terms as initialisms like USMC and SNP. So that's just my accent I have. And if you're more fond of saying SNPs and STRs, then I apologize. This is how I kind of tend to speak. I have a bachelor's degree in engineering and MBA from Rice University, Microsoft Certified Systems Engineer, been a DNA project leader since 2005, and I'm the founder of GeneticHomeland.com. SNP detection, biology, the human reference sequence, next-gen sequencing, things like that. So we all kind of know we're talking about nuclear DNA here, the different types of DNA. I think this group pretty well knows this stuff, the Y chromosome, and then specifically the male specific region of the Y chromosome, the Y chromosome structure illustrated here from 1 to about 58 million base pairs. Large portions are repeating segments that so far haven't been very useful for genetic genealogy. I decided to focus a little bit on the history of the genome because when we do these next generation tests, they're breaking the DNA down to little pieces, then they're testing it, then they're trying to apply it to a model of what it should look like. Understanding that that's what they're doing and that we're constrained to the model is important. So the Human Genome Project, of course, started with funding in 1990. The first sequence was about 85% in 2003. Celera Genomics used a different approach. Private industry sampled one man and got their first, they actually produced the first Y-DNA sequence in 2003 also. And so these human genome reference assemblies, basically periodically the consortium updates what it thinks is the master reference for human beings, which I tend to refer to as HG18, HG19 from 2012, HG38 from 2016. In the scientific literature, still most of it is reported and done with HG19. Subsequent analysis has shown that for most mammalian genomes, a single tiling path is insufficient to represent a genome in regions with complex allegic diversity. So this is kind of forward looking, but there will be multiple reference genomes in the future. HG38 is probably the last one where we say, okay, there's one master set. And remember that what drives most genome research is medical and biological studies. It's not genetic genealogy. They're not doing this for us. So most of the genetic genealogy laboratories that do the next gen test, they are now using HG38 assembly references. HG38, you can download the FASTA file. The Y chromosome portion of HG38 assembly, the human reference sequence, is actually composed of a mosaic, I think, of 3 specimens. And primarily those 3 were descendants of R1b, markers U152 and L20. So in the future, we have things like the telomere to telomere project, which is doing complete sequencing of one person and outside of R1b in many cases. So these are some of the GenBank entries for where they've done this whole end-to-end sequencing of the Y chromosome. They deposit it in GenBank. It gets an accession number there like CP086569.1. The overall thrust is that aligning longer read length sequencing onto haplogroup specific reference sequences is probably the biggest change that's coming in the next 10 years of Y-DNA genetic genealogy. So from biology to sequencing, I think you all know how to take a DNA test. Swab or spit into a collection kit, send it to the lab, receive a haplogroup assignment with a terminal Y-DNA SNP, and perhaps some matching to other test takers. Okay, we're going to just talk some details about the sequencing and the next gen shotgun sequencing. As I said, the DNA strands are randomly broken down into small segments, sequenced with chain termination reads, electronically assembled with computer software, and multiple sequences are run over the same molecule to produce randomly overlapping reads for a given position. So basically the way this works is that they can build a machine to read these small pieces quickly, and then the computers have evolved to be fast enough to reassemble it. Here's an example of kind of what we're talking about. So for a given set of Y chromosome positions at the top, here's 0 to 30. Each shotgun sequence might have a part of that. Sequence 1 has the first 10 markers. Sequence 2 has 15, 3, 4, 5 covers different. You assemble them together based on what you think the reference is, and you get the assembled sequence at the bottom. This, of course, is a simplified illustration. It has 6 reads, and it gives 2x coverage. At any one spot on the chromosome, there's at least 2 reads covering that spot. As I say, sequences are getting faster than Moore's law. The original Big Y, I think, was done on the 150 base pair read. Anyway, there are new machines now, PacBio RS2, 4,500 base pairs, Oxford Nanopore 6,000 base pairs, Ion Torrent 400 base pairs, which I believe you can buy a 400 base pair test from YSeq now. So increasing the sequence and read length is going to be the biggest change coming in the next 5 years of genetic genealogy. Part of what I wanted to put together in this presentation was to say, okay, this is what's coming. We're going to get more read lengths. We're going to get more data. How do we retain what we've already got in sequence, maybe from our uncles, brothers, and people, and integrate it with the new stuff that's coming? Because genealogically meaningful SNP mutations can occur at many points on the Y chromosome. Now, medically meaningful SNP mutations are located on genes. So that's a much smaller set of the whole DNA. But for genetic genealogy, we're really interested in any part of the chromosome as long as it's not recombining with the X. Our search interest is a lot broader than medical for the most part, especially when you start talking about STR, short tandem repeats. 250, 300 base pair read length is just too short to measure a lot of them. Sort of like trying to do a foot-long hot dog on a 6-inch bun. But there's also some evidence from researchers that next-gen at very high coverage may even be more accurate than traditional electrophoresis. So we're just going to do some work examples here, alignment of reads. So assume we have a reference sequence. So a reference sequence, which is what scientists say, this is what human beings should be, AGGTTCC. Our sequencer produces reads of 3 base pair lengths for sample one as follows: AAG, GGT, TTC, GTT, TCC. We have these raw reads, and an alignment software tries to figure out, okay, how could these reads match to a reference set? Here with this collection, there's only one possible way to do it. The first one covers the first three, GGT, the second set, and so on. So there's one possible solution, and the solution exactly matches the reference sequence. And this is just another illustration of that. It's perfect. All the reads map to the reference sequence. All 8 positions in the reference sequence have been found. Now, if we change the a little bit, some of the reads can only be matched at one position, but 3 of the reads cannot be perfectly mapped. What does this mean? If you look at, because what happened here was they changed an allele in some of the reads. When you align these, the best way to align them is that there was an A mutation from T to A at position 5. And if you accept that, okay, there was a change there, then you can get these reads to align to the reference sequence. And you have basically a single nucleotide polymorphism, T to A. But there were also alternate alignments that could have been done if we say, okay, there could be one difference from the reference sequence. And if we say, okay, there could be 2 differences from the reference sequence, now there's really a lot of possible alignments. So to get these reads to fit this reference sequence, we had to accept 2 differences just over a span of 8 base pairs. As we do alignments, do we want to accept that much? Do we not? How much change are we looking for? How much change can we accept? So it gets into, there's mathematics and statistics to it, but it also gets into kind of art and what is in a lot of times, I think, proprietary processes at the lab. There are alignment errors in your Y-DNA results, in my opinion. So here's a BAM view, and we're going to talk about BAM view more later, of an area that I think may have some alignment errors. This is around the HG38 position 10940385. And if you have a BAM viewer on your computer and you want to look at your own ref in this region, you certainly can. All these samples share a surname and numerous markers under RM222. So these people are related in the sense patrilineally, at least in the last few hundred years. But you can see they have a lot of differences by the color from the reference sequence. Most samples have 4 different out of 100 base pair reads, plus an insertion of 5 base pairs. So what are the causes for that? If it's just data, it could be that there's a pretty loose alignment parameters that were used. But it could also be biology, which is to say a large mutation or shift happened in a common ancestor of all these men that makes their DNA that they inherited different from the reference sequence. What's the solution? New sequencer equipment with longer read lengths will help, as well as improving the reference genomes that are used so that the reference is closer to what this haplogroup's answer is. So I'm going back. Right now, as far as I can see, nobody really deals with these issues. The labs kind of blow it off. The project administrators blow it off. Nobody's really addressing these. But as we start to try and move into more full understanding of the Y chromosome and we develop tools, these kinds of dilemmas need to be resolved. So real world alignment, billions of base pairs in the chromosome, 70 million on the Y chromosome, but it's one of the toughest to sequence due to repeats, palindromes, recombining subregions. So as I said, there's some art or craftsmanship in how various labs and DNA analysis services align the reads to make calls on SNPs. You can do your own alignment. The human reference genome is published free, and the alignment software is free. There's a Burrow Wheeler aligner, which was one of the first ones, and there's one called SNAP, Scalable Nucleotide Alignment Program. It happens to be my favorite. It outputs the BAM file very fast in one step. It was developed by Microsoft, UC Berkeley, UC San Francisco. It's built for Linux and Windows, which some of these tools aren't built for Windows, but this one is. So that's one I like, this SNAP. So tutorials on running your own alignment. There's a lot of tutorial material available. I can't go into all of it here. A lot of it's based on cancer research, and that's okay, because whether we're aligning for cancer or for genetic genealogy, the alignment process is basically the same. Some of the tutorials use Unix, Linux machines. If you don't run Linux at home, that's okay, because remember that you could rent a Google Cloud or AWS Linux machine pretty inexpensively and just pay for the hours that you actually use it. And then you shut it down when you're not using it. A lot of these tools have been ported to Windows and Mac. And so here's a link to an example of one, Introduction to Burroughs-Wheeler. That's I think a YouTube video. So I'm just going to give you kind of a high level orientation to some of these tools. SAM tools, it's a command line software utility for working with large next-gen sequencing data files. Most of the releases are on Unix, Linux, but one named Doug G did a simplified but stable version for Windows, which is one I use a lot. Okay, Ancient DNA. What I found is that when you do alignments with modern lab results versus ancient, you need to make adjustments to align the ancient, because ancient DNA broke down over time, tends to have shorter reads. So you typically want to run the alignment for shorter indexes and allow fewer differences. Map just illustrates some useful Y segments. Some types of DNA files when you start doing this kind of stuff. FASTA files, a text file, it just represents nucleotide sequences as character letters, C, T, A, A, C, 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 T. FASTQ is a text file for storing the reads and their quality score, and it's what comes off of sequencer machines. The SAM, or sequence alignment map, is a text file, very large, but it's basically a compilation of all those reads. The binary alignment map, because it stores data as binary at smaller size, faster performance. CRAM is a compressed binary data file containing only those reads which differ from the reference sequence. And a VCF, or variant call file, is a text-based columnar listing of only those portions in the sample genome where the reads did not match the reference. So if you take like an STDNA Big Y test, they'll let you download your VCF file for free. I think now they're charging $100 or so for the BAM files. Okay, this illustration of a FASTA file. It's a text file representing the nucleotides of each sequence. Typically, they're either 50 or 70 columns wide, most of them. So I've just kind of cut some lines here to show this is the HG38 reference FASTA file for the Y chromosome. Basically, what it shows you is that below position 10,000, they don't even try to make a call. They just use N for it. So it's definitely not telomere to telomere in HG38. FASTQ files are what you get if you look in online genetic archives. A lot of times what they'll have available is FASTQ. That's the raw reads, but there's no alignment. So that's where you're going to need to use some alignment software. They're often compressed with compression just to save disk space and transfer time, but there's some tutorials on how to download NGS data. And this shows an example FASTQ file from NIH's Sequence Read Archive. So part of where the FASTQ files are important is that if you go to Ancient DNA or scientific research papers and they say, okay, well, we're making our data available. Sometimes they'll make BAM files available, which have alignment, but a lot of times they don't. They'll say our data is available, but you have to get the FASTQ, and then you would have to run it on an alignment sequence to compare it to yourself or other folks. Okay, VCF, I imagine most of you are familiar with. And so some public DNA sequence repositories where you can get VCFs and FASTQs and sometimes BAM files. NCBI, National Center for Biotechnology Information. They're part of the National Institute for Health. They are the proprietors of GenBank, a bank of genetic information, and the Sequence Repository Archive. One thing about NCBI, when COVID hit, they switched a lot of their focus over to trying to work on the COVID pandemic and the genealogy information related to that, which is understandable, but they kind of turned away from some of the traditional things they did. So hopefully we'll see them start to do more work that helps us as doing human genetic genealogy. There's also the European Nucleotide Archive. That's a very good archive, ebi.ac.uk. It includes the 1,000 Genomes Project data. It also includes the Human Genome Diversity Project data. So those are international data sets where they've gathered samples from lots of parts of the world. Another good one, Seven Bridges Cancer Genomics Cloud, because it includes the Simmons Genome Diversity Project. The Simmons Genome Diversity Project, I think, was a privately funded project to try to basically get genetic information from all around the world, samples from all around the world. What's good about that project is that they they did it fairly recently, and it's got very high density of reads. So when you look at 1,000 Genomes Project data, a lot of times it might have space parts of it which aren't sequenced. The SGDP data usually has coverage of every position, certainly a lot more than the 1,000 Genomes Project did. It does require you to create a free account to do it, but for me, it's been worth it. The Estonian Biocenter is another place that has some sequences that you can download as well. So this is just a little diagram talk, because I get the question, because I publish a tree on Genetic Homeland and so on, people will ask me, well, how do I get the data in that research paper that said Viking, were this or that? Because I think I'm Viking, how can I compare myself? Anybody can do it. It is a little involved. This is kind of a general flow of it. Read the research paper, download the supplemental data and tables, download the raw data, the BAM files from the library, which is going to be probably one of the archives I had on the previous slide. If they have BAM file, then great. You can take that in a BAM viewer, compare it directly to your BAM file from STDNA or YSeq, and you can compare directly. And you can also use other tools. If they didn't produce BAM files, though, you're going to need to download the FASTQ files, run a software alignment, use SAM tools to do sorting and indexing to create your BAM file, and then you can directly compare. Right now, you've got mostly HG38 data from the labs. You may have old HG19 alignments from the lab, but someday the alignments are going to change. How can you keep your results up to date? The way you can do that is to extract the raw reads from inside the BAM file using SAM tools, and then realign them to a new alignment sequence using an alignment tool. And that way you can compare your sample with new samples that are collected in the future with other reference sequences. It's also the way you can move forward if you had relatives who are deceased now, who you have their BAM file, but it was HG19, and you want to look at it in HG38 or whatever comes next. You can do the same thing. You can extract the reads out of their old BAM file, do an alignment on it, and then compare it directly in a chromosome browser. Because the point is that reads are forever. Our global understanding of all the intricacies of human DNA keep evolving, and new reference assemblies will be created that account for all the unmapped scaffolds now. But if you or your testing company know how to produce a new alignment from your reads, you can always move them forward. If it sounds like too much for you to get involved in, YSeq does offer a fairly low-cost FASTQ mapping service, in this case mapping to the new CP0 86569 reference. I've talked a lot about BAM viewing, but now let's actually look at some of it. So we're going to use the IGV BAM viewer and view some samples. There are other viewers online, YBrowse, maintained by ISOG. FTDNA has a tool if you have the Big Y. And the 1,000 Genomes Project has a BAM viewer just for 1,000 genome samples. So as I said, individual reads are stored in binary files called BAMs. Here, we're looking at a G to A mutation at position 149.02414. So we see here that when each set of reads doesn't match what's expected, it kind of shows it with a highlighted A in this case. Down here, this G is the reference sequence. What we're looking at there is a G to A mutation in this individual. This is the same individual, two different tests from two different labs. One, I think, is YSeq and one is STDNA. So again, the way you do this is download your raw data BAM file from the testing lab, download the IGV viewer from the Broad Institute. What the BAM viewer gives you the ability to do is compare simultaneously multiple laboratories' results for the same individual, which is what we showed in the previous slide. But you can also compare for multiple individuals at the same time. One thing to note is that when using a BAM viewer, you often have to use a kind of an address nomenclature that is the assembly, the chromosome, the start position, and the end position. So HG38 is the assembly, CHRY is the Y chromosome. The starting position here is 2844421. And in this case, we're looking at 1 point. So the ending position is also 2844421. to use BAM viewers and some of these tools, you need to know the chromosome position, but what the labs give you a lot of times is just the name. That's why I created the DNA marker index on Genetic Homeland to be a cross-reference between all the known labels or marker names, such as M222, and what position they're at in each sequence. Okay, what a no-call looks like. Here on the right, we see a lot of reads. On the left side, there's 1 or 2 reads in blue. So this is kind of a thin coverage, and it may be a no-call in the results you get from the lab. Here's an example of comparing multiple kits: sample 1, sample 2, sample 3, sample 4. Say these are men, or maybe they're part of the same haplogroup, that we know they have a distant ancestor somewhere at least 1,000 years ago, but now we're going to try to sort out differences between them. Of the 4, the lower 2 have an A mutation at position 127904881. There you can kind of visually see that in the BAM viewer. We talked about this area before, where you have a whole lot of mutations or differences from the reference in a short space, one screen in the BAM viewer. There may be alignment errors going on, or there may have been a massive DNA change, but it's hard to make an interpretation. So now, talking about cross-reference at genetichomeland.com, I call it the DNA marker index, because we can search by marker name or by position. If we search by position here, we see that there's a marker called BY-27095. In this case, there's no assembly 19 position. Most positions are conserved from one assembly to another, but sometimes they're not. And so this one, we're not going to probably find it in academic literature because it simply didn't exist in that assembly. I think you all understand SNP versus STR measurement. Another way to think about it is the STRs tell you a house address, the SNPs tell you everything else: the continent, the state, the city, the street. So to get the full address, you'd like to have all the information. So here's one example of looking at some STRs in a chromosome browser. This is a section with CAG repeating allele, and the flanking sections are CCTTC preceding it and CAA after. The STR allele count will be the number of CAG motifs found between these two end caps. And if you count the number up, there are 19 CAGs in there: CAG1, CAG2, CAG3, so forth. And so your results on STR results would be, if this was DYSXXX, you would have 19 allele values there. We're going to talk about the nomenclature of SNPs a little bit. Basically, the position is, if we imagine the Y chromosome extended from end to end, and each base pair counted from 1 to 57 odd million, whatever that is, that's the position value. Now, as I said, in different assemblies, they put together different references or different concepts of what the Y chromosome should be. And so sections get moved, added, sometimes taken out. That's why the position is specific to the assembly. But we want to know our markers independent of the assembly. Thus, that's why labs started using these nomenclature labels like M269. Assembly 19, it was at position 227, blah, blah, blah. Position assembly 38, it's at a different one. But it has a T to C nucleotide mutation either way. Sometimes people think, okay, the SNP marker label, what does 269 mean versus 270? Does that mean 270 is higher or better? No, because they're typically just sequence numbers of the order they were discovered or recorded. There's no biological information normally in the 269 part. The prefix, the M, comes from whoever it was the authority or discoverer or publisher of that. So M is typically from Peter Underhill and Stanford University and associated researchers. Markers that start with PF from Paolo Francalacchi of the University in Italy. Because multiple laboratories and researchers might discover the same mutation independently, each mutation can end up with multiple names or aliases, or really they're synonyms. They all mean the same thing. So that's what my DNA index tries to do, is tries to enumerate all the synonyms so that you can go from a piece of literature to a person's home test to the national science databases. So RM269, M269, PF6517, and RS9786153, they're all synonymous. They're just different expressions or labels for the same biological phenomenon. A T to C mutation at assembly 38 position 205-77481. Indels, insertions and deletions. Indel is just a shorthand way of referring to insertions and deletions. It's sort of used as a term of art, pretty straightforward. I like to see insertions represented with INS in the ancestral value and deletions with DEL in the derived or alternative result. So here's some examples from Genetic Homeland, Z39589. It's a large 18 base pair deletion. Here's some insertions, a 10537. You see there the ancestral state is just INS because it didn't exist ancestrally. Mathematically, you could call it null, but anyway, it just evolved that the standard is to just put INS for insertion. The top one is an insertion of like 9 A alleles. The second one is just 1A, and the third one is a TGT. What do those look like in a BAM viewer? Here's the 39589, that 18 paired deletion. You can see how you've got these large gaps in here for 18. So that's telling you that those reads seem to be missing something. And insertion is a little harder to see because usually there'll be little icons like these 4s at the bottom of this, and you have to mouse over to see that it's a TTA insertion. On the genetic homeland table, you'll just see that the ancestral was in INS and the derived is TTA. This is an insertion of 4 base pairs. Multiple historical instances of a given, let's call it chemical change. Okay, those are represented with decimal suffixes. I think that's the best way to do it. Some people are using the whole haplogroup to distinguish, like IS7642 versus RS7642. And that would work if they only ever happen once in a haplogroup, but in fact, they can happen more than once in the same haplogroup. So for instance, S6932 has occurred in 2 different lineages in R1b. So what I think the best way to do it is to use a decimal suffix 0.1, 0.2, 0.3 to show which time it occurred. What this means, multiple historical instances, means the same chemical mutation at the same point happened independently at the same position, but in 2 different lineages. So it may have happened in one 10,000 years ago, and it may have happened in another lineage 1,000 years ago. And so the one here, S7642, one instance happened in haplogroup I, another in haplogroup R1b. You can also have another phenomenon where multiple positions for the same event. So this usually happens where there's a mutation that occurs on a palindrome, and then the whole palindrome shifts position in certain individuals. And so like here we have Z39589. Most commonly it's found at HC38 position 4571907. But some people have had a palindromic shift, and so that same 18 base pair deletion, you'll find it at chromosome position 4571871. In those cases where we're talking about a shift or the same mutation shifted, we use underscore suffixes. So _1, _2. So a phylogenetic tree, also known as phylogeny, is a diagram that depicts the lines of evolutionary descent of different species, organisms, or genes from a common ancestor. Phylogenies are useful for organizing knowledge of biological diversity. There's different types of trees that can be represented. It's all about finding ancestors. Trees can be done in many styles. A pedigree is a kind of a tree in a sense that it's the ancestral branches for one individual. This middle one here is called a radiocladogram style. This type is kind of a root and branch, straight lines, but they all kind of mean the same thing. They're depicting ancestry. You can make an ancestry tree potentially with anything. Could be the first name, could be the date of birth or something, but the kind we're talking about here are phylogenetic trees made from differences in SNPs. So we're making SNP-based phylogenetic trees. Basically, it's a tree of Y-DNA of mankind. And that leads us to something called the terminal SNP. we're going to talk about the growth of trees and some aggregate statistics of the trees and some sampling biases and characteristics. To recap, we talked about how to find and label individual SNPs. The phylogenetic tree from SNPs is about truth. It's not about probability. Here, we just compare the opening of the book of Matthew in the Bible. Abraham was the father of Isaac, Isaac the father of Jacob, Jacob the father of Judah and his brothers. In the same way, we could look at the phylogenetic tree and say a man with SNP B253 had a male descendant with a mutation we call M207. A man with M207 had a male descendant with a mutation we call M173. A man with M173 had a descendant we call M343. Okay, so that's what we're looking for. We're looking for unique SNPs that we find consistently on all the descendants of one person and on all their descendants. And from that, we can draw conclusions of who has it and who doesn't have that marker as to who their common ancestor was, at least in terms of these SNP mutations. We can't name them with names the way the Bible did, so we name them with various designations and labels for the genetic mutation that we can see. When we do the phylogenetic tree for SNPs, typically they're not consecutive generations. It's really an assembly of the mutations, and there are often hundreds of thousands of mutations observed. Only a fraction of those have been found useful for tree branching. So here we do a little exercise with trees. We have 4 markers tested. We have 4 people tested. Person 1 was positive for markers 1, 2, 3. Person 2 for 1 and 2. Person 4 for positive only for marker 1 and was negative for markers 3 and 4. Can we make a tree for these 4 people using these 4 markers? Yes. And that tree would look like this. Marker 1, everybody has it. Marker 2, only persons 1 and 2 do. Marker 3, only person 1 has it. Marker 4, only person 3 has it. If you put that together, then you could get a tree that looks like this. Person 4's terminal SNP would be marker 1. Person 3's would be marker 4, and so forth as illustrated here. So using 4 markers, 4 people with differences, we're able to make a tree. So the terminal SNP refers to the deepest SNP marker on the tree that an individual has tested positively for. So as of a couple of days ago, we had 31,920 distinct terminal markers on the Y-DNA tree. If we extend this exercise and we test more and more markers so that everyone has a distinct difference, meaning they have a marker that nobody else has, and that's what the Big Y and the Next Gen test are doing. They're extending the marker counts from 111 STRs to 20 million, 11 million or more SNP markers. So now we're able to get much deeper trees, much deeper branches, and to get a unique SNP for just about everybody, and certainly for every family group. So this is how it really should look. If we tested more markers, we found marker 5, only person 2 had it. We found another marker 6, 3 and 4 had it. We found another marker 7, only person 4 had it. So now all these people, each one has a distinct terminal SNP, person 2, person 3, person 4. So their terminal SNPs changed. Their biology didn't change, but what we knew about it and how good of a sample we took of the biology is what changed there. The phylogenetic tree is growing because genetic testing is growing so much. In 2002, this was the chart of the tree of Y-DNA, tree of man. It had 18 letter haplogroups, where I would have been on this tree all the way from the ancient to now would have only taken 10 steps. So 10 branch levels. 2002, that's 20 years ago. Okay, today there's about 70,000 branches on the entire Y-DNA tree. There are 2.3 million marker names or labels, of which about 2,029 are mutation events, meaning taking away the synonyms. And it's been growing at the rate of roughly 750 new branches and 15,000 new SNPs per month. And that's been pretty steady. This is a histogram of a branch count of the tree just in a couple of years by haplogroup. For instance, here on the right, for haplogroup R, in 2019, there was about 9,400 branches. Today, there's over 27,000. So this shows you there's been growth in a lot of them, some of them more than others. The some being the ones that are most common in Western Europeans and Americans, and who get their have gotten a lot of testing done. So they're in haplogroups I, J, and R primarily, and E a little bit. So at Genetic Homeland, we post a summary page of how many branches and markers we found in each major haplogroup here, just for general information. Part of the reason we do this is because our focus now is on trying to build out and collect the trees for all the other haplogroups. So, because what we're really doing is trying to establish the tree of mankind. And so we're trying to make haplogroup O grow because a lot of Chinese are haplogroup O. We know that should be bigger. And so this kind of counts our progress. The tree backbone, when they were first grouping and typing people into clusters, they couldn't know exactly who was the most ancient. So they started trying to make it kind of alphabetical sequence, A and then B. Let's zoom in here. A and then B, and then they had some C's and D, E. But then at some point they realized that, well, actually people in haplogroup P, which includes descendants R, are actually descendants from this section K. It turns out that haplogroup R is what a lot of Western Europeans are, is actually a descendant of P, which is a descendant of K, descendant of F. So in some cases, like this just have to kind of know the backbone of the tree, I call this, if you're doing the whole tree. There are about, by my count, 47 backbone branches under Y atom that make up the 23 major letter groups. Another thing we have on Genetic Homeland is a pedigree tree to show you as an individual, your ancestors by the Y-DNA tree. So it's not showing all the side branches. This is just your branches. So in 2008, this person had 43. Okay, in 2022, now there are 56 branches that we know about. So that's how much the tree's grown, 13 levels. Point here is that when we think of M269 and we get that first 13 marker STR test, it all told us that if we were, maybe we were M269 or M253. That right now, M269 is at tree level 29, and it's 29 levels below Homo sapien. But look how many more levels there are. So this is a key point, especially with new people or people that don't understand. They're getting kind of a false sense of information from knowing they're M269. To really say that somebody's fairly closely related, you probably need to be at chart level in the high 40s and maybe even to the 50s to say these people down here, like I know in this chart, to have people that only have the same surname, you have to be at level 55. That's one way I like my pedigree tree, which is I can immediately tell how far someone's gone in testing. If they're only in the 30s, but they think they're related to other people in the 30s, it's like, yeah, you are, but I mean, it was getting close to the Neolithic or whatever. It's just a tree to let you see what your Y-DNA ancestors were. It is printable, so you can print it out in about 2 or 3 pages. Tiger Walsh made a nice picture here where he shows the giant tree of mankind's lineages. And that's a kind of a nice way to think about it. It's not the autosomal across all the chromosomes. The Y chromosome is just 1/23rd, but it gives you a true view back. So this is just kind of saying somewhat I was saying before. Branch level 40 is roughly 2,200 years of ancestral time. This marker was only discovered 16 years ago. So it's a process of ongoing discovery every day, every week. That's why the tree's growing so fast. Coincident mutations, groups of mutations which always coincide together, meaning every time we find somebody that has one, they'll also have the other. They're useful in phylogeny because when we find one of these markers, we can usually make a branch assignment of that person. So how coincidents are displayed, like on the Weifel tree, here, this is a branch A7135. To the right, it shows coincidents such as BY5267, Y18358. When there's more than 3 coincidents, they have kind of an abbreviation. So basically, what the coincidents also mean is that on the phylogenetic tree, they're going to be equivalent. Like each one's going to be at that same branch level. Here's how it looks kind of on the STDNA Y public tree. You navigate to your branch node, in this case, EA7137, change the view to variant. And these rows will display the +6 is a numeric count, but then these other markers here, like A7136, BY153285, those are coincident to A7136. So where you find one, you'll find another as long as the tree is correct. If you find two people who don't have them coincident, that means we need to add a branch to the tree. So that's not a bad thing, that's a good thing. So the biological causes of coincidence are that one ancestor individual got 2 or more SNPs from his or her parent. So you could inherit 2 SNPs in one generation, but more often they're an accumulation of mutations across multiple generations, combined with a population bottleneck such that when we see one, we see the other. The other reason we can see coincident mutations is because we have limited sampling. We just haven't tested enough of those descendants to break out which one they are. So that's often what we see when we go to like populations that haven't been seen very much, such as say a South Pacific Islander. We'll find many mutations from the human reference sequence, but we only have one person, one instance. So we can't put those into multiple layers. They all become part of one branch at the time they're discovered. Eventually they get broken out. Coincident mutations can be very helpful because it's rare, but where we do have a back mutation in one base pair, we can still look at all the other coincidents and say, okay, if they have 9 out of 10 and that one back mutated, then we can either add a branch to the tree or we can say this is just a back mutation. So from a tree building perspective, and if you're working on your recent Y-DNA cousins, that's what you're trying to do is build trees. For recent people, the coincident mutations are kind of like stardust. They're the dust from which the next branches will be born. So in this pedigree, at this time, BY21680 was the terminal SNP. That means that's the deepest SNP marker on the tree that an individual has been tested for. It gets used a lot, but it can be confusing when it changes. It can change when we take a new YDNA test and we get more markers tested. It can be changed when results from existing tests get reevaluated thanks to expansion of the phylogenetic tree, meaning other people get tested. I think of it as the most unique SNP marker that's visible at a moment in time. Your DNA is not changing, but the parts that we can see are changing, and it changes when you get an upgrade at the lab, more persons provide samples, and the phylogenetic tree expands around you to derive sequences of related individuals. So Big Y at STDNA has a matching feature independent of surname. Novel variants is a feature such as Big Y. These are your unique ancestral markers. They're the name that has been changed from. They've also been called unnamed variants. These are your discoveries. We should celebrate them. As the global phylogenetic tree grows over time, these variants will become named SNPs, and a lot of them will become phylogenetic tree branches, your tree branches. Over time, the number of unnamed variants that you have should shrink. I think when I took the first Big Y in 2014, I had 78 novel variants. Today, I have none. That's because all those variants have been identified and broken out into branches amongst various Y-DNA cousins. We're going to talk some more about using the Big Y tree at STDNA and Big Y samples. There's some limitations there. So these are kind of some of the tricks to know. The novel variants in chromosome browsers have some limitations and discontinuities. For instance, searching by position doesn't work unless that position is part of what is listed in your private variant. You can't just search for any position. The novel variants they show a lot of times already do have SNP marker names associated with them, but for whatever reason, because it's probably 2 different pieces of software and systems that run these processes, they don't work. So in this case, position 1237023, I'm looking on the bottom right, had a G to A mutation in this case. It's listed as a novel variant, but actually it's also been already labeled FTC 72154 by FTDNA. So a little bit of the left hand and the right hand not quite connecting yet. Another thing that happens is that a search on a marker name comes back empty, even though that position is listed under the novel variant. So if you want to see it in the chromosome browser, you sort of can't, because when you search for this marker name on this result set, it's going to say no result. So that's weird because it also told you there were some. So there is a workaround. If you can use a tool where you can search by a range of chromosome positions, such as Y-Browse and Genetic Homeland's DNA marker index. You can see other names kind of in that region of DNA. So this one we're talking about here in this real world example, FTC 72154, there's a marker 719072 that's located roughly 20, 30 markers away. This is sort of the same process on Genetic Homeland. You can enter a range search and return all these SNPs along that range. We found Z19072. We try that in the search tool and it says, oh, okay, we have that one, not required derived. So we open the FTDNA chromosome browser with Z19072. There's no change there. See, it's all just whatever the reference is. But if we scroll it back to that position, 1223703, we can clearly see, yes, the person does have the G to A mutation there. There are workarounds, there are things to do, but if you're trying to put together a YDNA project or something, you pretty soon realize that, you know what, it would be a lot easier to just get the raw data, use a BAM file and other third-party tools on your own computer time. So therefore, the best thing is to just download the raw data files. Another thing you can do fairly uniquely with the DNA pedigree tool at Genetic Homeland is you can compare any two terminal SNPs from anywhere across the whole tree, and it shows you where they diverge. So when you're running a project and you have this person has one terminal SNP and this other person has another, and maybe it's just changed, and the first 4, 5, 6 you can kind of remember, but now that we're like at 50 and 60 terminal SNPs in a FTDNA project, nobody can remember that on their own. So therefore, this tool lets you just do a comparison, in this case, between BY21680 and 8725. Okay, so what you see is they were together at M222, but then they diverged here at level 41. The person on the right has a marker 35297. The person on the left has S658. And so you can see the divergence there. So that's one of the good things I like as a project administrator with this tool. It lets me very quickly tell not only the level of the divergence, because again, if they're closely related, it should be in the 40s or 50s, but also let's see the path. Shifting gears a little bit. So we started out talking about definitions of SNPs and how to do alignments and things. Then we looked at groups of SNPs and who has them and who doesn't. That leads you to trees. Most people are interested in their geographical and their ethnic attribution. They're not really interested in all that stuff. That stuff is just how we try to answer their questions. Because let's face it, everyone wants to know, I want proof that I am an Antarctican. I want to know the marker that tells me I have been right all along in my research. It matches what my grandmother told me. It shows me that I am royalty. I'm sure you all have heard similar inquiries. So my way of looking at it is to be cautious and open-minded and to realize that humans have always been mixing and traveling. For every marker where we conclude that marker XYZ originated in a specific geography, there are exceptions, but where we see a lot of geographical continuity in a lineage, which is well sampled, it does suggest a geographical and cultural heritage. The challenge is that usually you need much higher resolution than what the Y37 or whatever people did with STRs, and they're trying to draw conclusions up. So kind of a phenomenon I call a Mountainous conclusion out of a molehill of data. They need to get the big Y, they need to be in a group that's well sampled, meaning that they'd have very few novel variants left, and then we could start to make some inferences. The Viking study that was done by Margarian in 2020 was a really good one. They collected 297 Viking grave samples that had Y-DNA that was able to be measured. They know they're Viking graves because of the stores and specific metal, objects that are in there. So they have a very clear-cut sort of physical remains from an archaeological standpoint. So we know these people were Vikings because they had Viking gear in their grave. And so when we looked at the DNA, we would have thought that they were all eyes because people had the presumption that eyes were most common. But what we also found was that eyes were most common at 32%, but R1b was also common almost as frequently at 29%. They also found 8 haplogroups that are at low frequency today, L and T and Q1b. Kind of leads you to the conclusion that even 1,000 years ago, Viking was an occupation almost as much as an ethnicity. Using the STRs and trying to derive cultural inferences from that is what I call low resolution fallacies. They're just too low of Y-DNA resolution. The ethnic attributions to these, to some of the common markers like M269, M223, L21, those happened in the Neolithic and Bronze Ages, if not earlier. So they're meaningful at the continental level. Meaning if you have L21, your ancestor came from Europe, didn't come from Africa, didn't come from East Asia. Okay, that's true. But what most people want to know is what county in Ireland are they from and what city and what county, so that they can do traditional genealogy research. For that, you need a lot higher resolution and better sample collection. So by the same token, we also see that genetics attributed to historical events and populations are often vastly oversimplified. So we already gave an example with the Viking study. This is just a look at what the Y samples are for ftDNA overall. R is 45%. This is a notional world map of where pre-colonial Y-DNA major haplogroups moved. So like Native Americans, almost all Q, a little bit of C, and so forth. Today, these are, in the modern populations, these are thought to be the most common haplogroups in the different parts of the world. But again, it's just a concept. There's many exceptions. So when people ask me this question, does this mean I'm Irish, or does this mean I'm from County Tipperary, or whatever it is, the way I like to look at it is, okay, how high resolution is your DNA result? And then how well is the population you're trying to compare to tested and sampled? And so that led me to an analysis about sampling penetration by country. The number of matches we have can be a function of how many other persons from your group have been tested. So what I tried to do was count samples by the country, attributed to origin, and then plot that. So this is, again, a histogram of the Y major haplogroup count at ftDNA. I shouldn't say that. This is a branch count, which is going to be roughly proportional, but this is the branch count as of 2009. These are the most densely sampled countries according to Y-DNA ancestor and attribution. So if you take how many samples we have that are attributed to that country and divide it by the population, that gives you kind of a sampling density. And so this is a sampling density per 1 million of estimated population. So Ireland, 1,656 Y-DNA samples per 1 million population. Pretty high, higher than most other places. Finland, 446, United Kingdom, 346, Norway, 294. Look toward the right, places you might not know about: Saudi Arabia, 125, Qatar, 124, Mongolia, 141, Denmark. Germany, you'll notice, is one of the lowest on this group, which is important to remember in that there's so much German heritage that came into the colonial United States, and people take their tests and they want to know, where's my German? Part of the challenge is that we don't have enough samples from Germany itself, from people, lineages that have stayed in Germany, to have a good sampling context, I feel. And that's also true, I feel, on the autosomal side, because myself and my wife, we feel our German percentages are vastly underrated, because again, we feel like there's probably not enough German samples in the autosomal databases. But anyway, that's just an opinion. These are hard numbers, not really too debatable. So how complete is the Y-DNA phylogenetic tree? We're getting very complete in haplogroup R1b. We have sample coverage of over 400 samples per million in the British Isles, but we still have very low coverage in Asia and Africa. So as an exercise, estimating ethnicity using the tree. Look at YFOL tree for YF536. There are 2 samples. One is from China and one is from France. This is kind of a curious branching. How could that have happened? If I had Y5362, does that mean I'm probably, Chinese or French? If you didn't have any preconceived notions, you might wonder. So the phylogenetic context helps inform that. If we zoom out a little bit look at the ancestral, the sibling, and the descendant branches of this branch, what we'd see there is that the samples are pretty much all European. If we look at the descendant branches of 5362, again, they're all European, they're not East Asian. It's likely that the Y5362 is a European origin and somebody had a recent introduction into Asian that tested that sample. Now that we're doing all that, of course, without interviewing the person, taking their history, doing traditional genealogy, that would all inform that. But looking at the ancestral and sibling branches and the descendants, if there's a lot of them, that gives us some idea of what is the likely ethnicity. European, not Chinese in this case. To do that, there are multiple tools. STDNA has a nice country report, which gives a tally of national attribution for markers and descendants. What's good about it is it shows all downstream participants. That can be good. When people want to know the mutation age, I personally am much more interested in getting the sequence of mutations right and getting the pedigree in the right order, but YFOL does do a good job of trying to put together some aging. So the phylogenetic tree tells us who our paternal ancestors were, and it lets us compare one person against another. No percentages involved, it's straight truth. And the next gen Y-DNA tests give you thousands of SNP results to help clarify the issue. To connect paper genealogy and genetic genealogy usually requires numerous DNA project participants who had next gen tests and deep paper pedigree for at least some of the test takers. So the way I like to think about it is flipping the script. Rather than thinking about, I have a surname Jones, that must be my paternal ancestry. Instead, consider thinking about, I have SNP markers A, B, C, and D. Those define my patrilineal lineage. People ask, can you give me contact information or help trace my genealogy with Y-DNA matches? Privacy regulations prevent revealing information not already provided by the testing laboratory. So my best answer is to get the most complete Y chromosome test for yourself and recruit samples from others from the geography, the surnames, or other areas that would be most informative for your interest. In other words, the answer to your question probably lies as much in collecting other samples as it does in your own, part of what underlies my ancestral parish sampling. Also try to sample all your paternal lineages and watch out for low resolution fallacies. Royal descent. Most celebrities and royals are not fond of revealing. Myths in family genealogy are not uncommon. Low resolution markers can be useful for disproving a relationship, but they cannot prove one. So RM269 is shared by hundreds of millions of people. You really need high resolution SP and SNP markers. We talked already about how some of the limitations with the STDNA chromosome browser on their site can lead one to want to download the files. Unreliable mapping, indel versus a sliding point mutation, especially as you look at older mutations. In this chart here on the right, you see we've got representing positions 1, 2, 3, 4, 5, 6. The reference sequence for those is AGT-CGG. The biology, what happened is that there was a deletion of the C at position 3. So the resulting biological run of the DNA will be AGCGG. That's what you will measure when you read them. So in terms of what labs do when they call it, do they recognize it as a deletion at position 3? Or do they record that, well, there's a T to C mutation at position 3 and a C to G mutation also at position 4? So either result is possible. So the bottom line is labs do a good job of trying to overcome that, but there are spots in the DNA, especially in some of the haplogroups outside of R1b, where we can see that happen. Faux mutations relating to assembly 38 reference sequence. This happens when a mutation occurred ancestrally in one of the samples used to compose the assembly 38 reference. And the people that were used in that reference were part of haplogroup R1b, and specifically SNP L20's lineage. All samples outside of that lineage would be expected to show a derived allele at HG38 position 6154452. They would show it as a deletion. But it's really a faux mutation because what actually happened is that it's an insertion at that point, ancestrally in haplogroup R1 ancestor. These are most common for newer laboratories that are relying entirely on the human reference sequence, such as 23 Mofang. They have tons of faux mutations. So we try to note those on the DNA marker index as saying, it's probably a faux mutation. And the HG38 reference is not ancestral, meaning it doesn't represent the ancestral state. So just keep that in mind. This is an example with MF181966, described as a deletion by 23 Mofang, but through wider sampling of people in R1b, R1, R2, and other haplogroups, we see that really there was just an insertion in that one lineage, but that lineage was used to form the human reference genome. When working outside of haplogroup R1b, use a position searchable tool to look for other markers listed at this location, such as genetic homeland's DNA marker index, ISOG's Y-browse tool. Confirming with a BAM viewer, this is an image with samples that are across many haplogroups, A1b, B2a, E1b, C3, F2, H1. There's a kind of a cross-section of humanity there. And you see that in every case, it looks like a deletion on the chromosome browser because the chromosome browser is going by the human reference sequence at the bottom with that A there in the purple circle. So that's what it looks like in a chromosome browser, but really ancestrally by the analysis and using the phylogenetic tree, we can conclude that really it was an ancestral insertion. Okay, palindromic regions, palindromes, same forward as reverse. DNA palindromic sequences tend to chemically attach to themselves in a loop, and these loops can cause large insertions, deletions, and shifts, creating palindromic arms. Palindromic arms were probably more problematic in the STR era of genetic genealogy because they affected the counts of the repeats. Because next-gen sequencing is done from cutting up the strands into small pieces, the whole palindromic arm position is not as important, but it could become a bigger challenge again when we go to longer read length. This is kind of a nice ideogram that Thomas Cron did some years ago about palindromic arms on the Y chromosome. He also noted from a study that the Y chromosome has more palindromes than any other chromosome. For instance, chromosome 1 was estimated to have 2.6% of its length in palindromes, chromosome Y 13. It's part of why the Y is so challenging. XY crossover recombination. In our idealized world, the Y chromosome part doesn't recombine with the X, therefore it's always a true signal of ancestry. All the mutations we see must be things that happen in some ancestor. But there are pseudo-autosomal regions, PAR1 and PAR2, where we seem to see some recombination with the X chromosome. Some researchers believe these should not be relied upon for patrilineal ancestry markers, but other labs are reporting and using these mutations on their trees, and there's quite a lot of them now. So they seem to be working, so we have them on our tree too, at Genetic Homeland, with the caveat that we have a note. If you look in the marker index, it'll have a note in there that says it's located on this region or a heterochromatic region or whatever. Some relative strength of the various published phylogenetic trees. You could say, okay, which tree should I use? I guess the short answer would be maybe all of them, but here are some of the relative strengths and weaknesses as I see them. FTDNA has the most Y samples, and they're growing. Their public haplotree has a good public linkage of surname, country, and Y-DNA tree branches. They have about 60,000 branches on their tree at this time. Their customer base, of course, is skewed toward US, Western European, and Middle East. So they're best in R1b, I, J, and E1b. They have a lot of low resolution legacy data, however, with customers who only have STR results. But we do continue to see upgrades pretty steady, so that's good. YFull has the oldest well-maintained public tree, about 28,000 branches at this time. It's not a lab, but rather a results analysis service. They have superior collection of R1A and Asian samples because they are located in Russia, and they incorporate a time origin estimate at each branch. The ISOG tree was used by the scientific and academic community. Tree and tools have not quite kept pace though. The last full update, I think, was 2018. It had about 9,700 branches on it. YSeq has an internal tree where their product development's gone. I'm not aware of where it's fully exposed, but it can be the best in niches because where they have a lot of customers, such as under M222, their tree could be the best in that niche. But in general, they use the Y-fold tree for their broad classification. 23MoFang is the largest East Asian laboratory with a lot of samples in O, C, and Q. It's in the People's Republic of China. It's not easy to navigate the site as it's not published in English, but they have about 26,000 branches on their tree. And then Genetic Homeland, the site that I run, where my goal is to try and have the biggest, best tree on the planet. So we have about 70,000 branches, and we try to integrate all those other branches to make the most comprehensive tree of mankind. Here is just a visual graph of the relative size of the branch counts. So as we said, there needs to be a comprehensive cross-reference of SNP marker names, as well as the positions with the different assemblies. The DNA marker index on Genetic Homeland is one. There is an older tool called DBSNP, which was around but isn't as focused on genetic genealogy. So some of the features we have on Genetic Homeland, we cross-reference the SNP labels across all labs, across all chromosome positions. We have the largest SNP name cross-reference, 2.4 million labels. We integrate the trees with scientific research papers as well, 70,000 branches. It's intended for individual pedigree and head-to-head comparison for divergence of two markers. It's not designed to be a bushy tree display. The others do that much better. And generally, we do not have specific samples for each branch. We do have mapping of geographies for all descendant branches for a given terminal SNP, including an ancient Y-DNA data set with over 4,800 records. So you can compare the geographic distribution of up to 3 different terminal SNPs at the same time. So that's something that I don't think you can do with the others. You can compare the distribution, and also it will automatically look for all the descendant terminal SNPs branches and put that into one graph. So to recap, we talked about SNP detection, trees, and interpreting SNPs, and some tips to kind of the topics. So a little summary of the sequencers and where they're going. Again, increasing the sequencing read length is probably the biggest change we're going to see in the next 5 years of YDNA genetic genealogy. And we'll see multiple reference sequences, probably to the major haplogroup, and then probably to specific big nodes such as R1b DF27. There's a lot of descendants of that, U106, M222, so forth and so on. Aligning longer read length sequencing onto haplogroup specific reference sequences is going to be the next biggest change in the coming 10 years. So now we'll open it up for questions.
Brad has graciously made his PowerPoint slides available, and I'm including them on this slide so you can download them